now it's time to go back to the second half of Professor Raed's talk.
It's on the different.
Okay, the second part of this presentation, I'll discuss the interaction or the effect of GcMAF on nagalase. Now, some people have, I don't know how to say it, but some people can say these words easier than other people. Scientists tend to complicate sometimes the nomenclature of the terminology here. Alpha and acetyl galactose aminidase. I don't know if you can do it faster than that. So in short, this is nagalase, which is an enzyme. Nagalase has been around for years, not just now with GcMAF, but it's been there for years because you normally produce it. In chemistry, they say that this is a glycoside hydrolase, and it clips or removes sugars. Glycoside means a sugar, and it removes sugars from an organic substance. And this is normally produced by humans and bacteria. Now, you'd think from the presentation that nagalase is something unique to cancers or unique to viral infections. We tend to see that there's accumulation or there's increase in the concentration of nagalase in the blood of cancer patients and viral diseases. And of course, this increase also will affect the GC protein itself because it will result in the removal of the sugar, or what we call deglycosylation. And if that happens, you will not get GcMAF, so it becomes useless. And this, of course, will not activate the immune system, so there is an immune suppression happening. You've seen something similar to this in Professor Marco's presentation. This is the protein itself, and then this is the important one, the amino acid 3-onine. And here you have these sugars here attached to it. This is the vitamin D binding protein. And sequence-wise, these two branches are clipped during a normal, healthy immune activation. So beta-galactosidase produced by B lymphocytes removes the galactose. So as you can see, galactosidase and digestive removes galactose. And then T cells or T lymphocytes, these are two components of the immune system. They remove the other one, sialic acid, because they produce alidase. And now suddenly you have GcMAF. This is GcMAF, the active compound that will activate the immune system. Now what happens if you have nagalase? As Professor Marcus said, nagalase will not do anything to this one. Nagalase can act on this one and eclipse this covalent bond or this one. It will cut this one. So it goes here, cuts this one, and then suddenly you have just a protein, threonine, and no sugars attached to it, no sugar. That's why we say it's deglycosylated without a sugar. So this is what nagalase does. Now, you see that we have the protein, the original backbone of the protein, but we don't have this sugar component that's important for the function of GcMAF itself. Okay. Now, as I said, nagalase itself is normally produced by all cells, and you need it. And people who don't produce nagalase or who have the genetic deficiency of nagalase, they develop something called Schindler's disease or Kanzaki disease. So you need some of this nagalase for normal functioning of the cells. Okay. This is a rare congenital autosomal recessive disease and can result in metabolic syndromes and accumulation of some sugars in the body itself. And usually this is fatal when there is complete deficiency of nagalase itself. The gene for nagalase is called NAGA, and it's present in humans in chromosome number 22. And as I said, if you don't have this gene, or if this gene is mutated, malfunctions, then there's accumulation of sugars, glycosphingolipids in the body, and this is toxic and can be fatal. Okay. Now let's look at what happens to nagalase and its interaction with GcMAF. In a study, 20 advanced cancer patients, blood from 20 advanced cancer patients from Europe and the United States, they were tested for nagalase before and after the use or the administration of GcMAF. All of them, at the beginning, before giving GcMAF, they had high nagalase level, 2.84 plus minus nanomolar per minute per milligram, which is way above the normal concentration of GcMAF. Now, after giving or administering GcMAF following weekly injections, almost all of them, except for one, had significant decrease in nagalase. And now we have 1.59 nanomolars per minute per milligram. And this is statistically significant, as you see, p-value is less than 0.01. So there is a significant decrease in the concentration of nagalase with the use of GcMAF. This is in vivo in the patients themselves. The other thing is that, nagalase decrease was also associated or linked to also improved the clinical outcome of those patients, clinical condition. As you know by now, no adverse or side effects were reported using GcMAF because this is a normal protein that you have. Now, where do we go from here? Probably we need to try this on different disease states, different cell lines. Probably, and this is what I'm trying to do in my lab, looking at nagalase activity, molecular level, like using real-time PCR, and also at the same time secretion outside using protein estimation. The sky's the limit here. Lots of things here can be discussed, lots of ideas are there, and probably we need to have more in-depth look at what happens when GcMAF interacts with, for example, tumor cells and the sort of signaling that you see. The same thing applies for the first part of the talk with PV2 cells. Now, as Professor Marcus said, they're going to do microarray studies to look at hundreds of genes and what happens to them. We can do the same thing and see for all cells, different cell lines, different cell types, what happens, and which genes are upregulated or which genes downregulated. we can also look at nagalase and its effect on other aspects of the immune system or other disease states, not just cancer. Maybe it's also present in high concentration in other disease states. So we have lots of things in mind, and the future looks promising, inshallah. Thank you very much.
Thank you, Raed. Are there any questions? Yes.
Thank you. Steve Hines from Texas. Have you made any correlation between oxidative stress and the levels of nagalase or GcMAF, and particularly serum iron or ferritin levels?
To the best of my knowledge, personally, no. But to the best of my knowledge, this has not been done. At least not in the published literature. I haven't read it.
Are you aware of any assays of infected macrophages before and after nagalase to see if after activation of the macrophage, does it cause the intracellular infection to be resolved? I'm referring to macrophage-based infections for those of you who may not understand that.
Well, if the question, have I done that or worked on that, the answer is no. I haven't done that.
And not aware of anyone that has.
Excuse me?
You're not aware of anyone else that has studied that? Okay, thank you.
Are you doing nagalase testing at the University of Sharjah, or are you planning to do the test if you are not doing that?
Well, I don't know. Probably maybe David or should answer this question. Yes, I'm planning to do that. We are in the process of doing this. There is a new test here for nagalase, and there are we need to, we need other equipment as part of the assay itself. But yes, it is there. We're working on this as we speak. I mean, I have samples in the lab and from different disease states. I collected samples from viral diseases, and we're getting samples from tumor, from cancer patients. And yes, we are planning to do something like that, yes.
Why has it been done before? Is it hard to do the nagalase test? Why has it been done before in our labs? Here in the UAE.
In the UAE, you can't even generalize. I don't know if there are many places that will do nagalase test even worldwide. Very few places do that.
I know in Holland they do that. Is it the United Kingdom they do nagalase test in UK?
We've developed a new test with a completely different technique for alpha nagalase. There's a urine nagalase that's not relevant at all. It has to be the blood nagalase, the alpha nagalase. And we may be putting it into production this year. And it's very likely that Professor Raed will also be doing it over here.
Thank you.
So it is in the pipeline, in other words, that's why.
Thanks.
Yes.
Do you think nagalase levels would be specific to races? Different, say like population of Indians, or will it differ?
Different ethnicities, you mean, or that's what diseases? You mean in healthy people?
Yeah.
Well,
See, like if you compare it with, say, a family having a patient with cancer, have you tested with the family and compared it with the patient? And in the interest of autism, have you compared the level of nagalase in an autistic child with a sibling or the parent? Is it different? Yeah, because that might throw more light on the photo.
Thank you for your question. I think Dr. Bradstreet knows a lot more about babies and children, autistic children, and parents for grandmothers and family studies. But yeah, he did this work, and it sounds of correlation as far as I know, but he will speak tomorrow. So maybe you ask the same question again, but maybe you can answer.
But I think one remark here is that ethnicity-wise, this is a different story. I mean, families and children, autistic children and so on. But your question here is, if we go to Southeast Asia, is Nagalese different than other parts of the world? Is that what you mean?
Yeah, it is both. The ethnicity and also
Why would you think that will be different? I mean, this has normal function. Baseline Nagalese should be similar, I think.
we are not done that.
I mean, no at all. But it's interesting. But this comes down comes back to the previous question, and that is why Nagalese testing is not widely spread. I mean, if you search for a kit to measure interleukin-1, you will find 100 companies with 100 kits, and you can buy them. But there isn't, to the best of my knowledge, a kit that you can buy from the market to do Nagalese, at least for the time being, to do Nagalese in your lab. So what usually happens is that probably the existing one, before what David said, you have to send your samples there. Isn't that?
Well, we should just point out that Odeh has a company in the Netherlands called ELN that does Nagalese testing. And we send all our Nagalese testing to EMA.
Do we know any physiologic response that seems to create an increase in Nagalese? And could the need for cells to use more sugar be the reason why that enzyme becomes more necessary? Maybe infection stimulates the process in which Nagalese becomes necessary?
Oh, well, in the literature, I think I read something about that, but I don't remember the details of that for time. No, yes, there is something about that in some different disease states, and what it does to sugars, and it's linked to other mediators. But
on doing the ketogenic diet, do you think there is an impact with Nagalese levels doing a ketogenic diet? You know, we talk about sugar and the isolation, can't say that quickly. So could Nagalese be affected indirectly by dietary strategies? It's particularly like a hyperketogenic diet.
yes, there is this type of research. I think was done years ago. Yes, and there is interaction. There is an effect. Yeah.
Is that it's a good effect?
Yes.
Excellent.
Yes.
In short remark, that is, if you get the flu, yes, then within a couple of days, according to Yamamoto, the Nagalese is increased. And it is increased up to 3 months. You have a flu,
Yes.
virus infection.
Yes. Viral disease. Yes.
So that is maybe causing some problems in the interpretations.
Stephen.
Thank you a lot for your very interesting talk. I'm just a normal doctor, not a scientist like you. But are there any data on the treatment of reticulosis? I only find some things about Carter disease, nothing about Hodgkin or leukemia. Another interesting field is, of course, children's cancer. I never read anything about it yet. Do you know anything about these?
For GcMAF you're talking?
Yes, treatment of GcMAF in children cancer or in reticulosis.
I think I think we will have presentations today or tomorrow.
Yes. Although we have 3,000 autistic children on GcMAF, we have almost none, no children at all with cancer. And you wish to say something about that?
That exists at least. And it's very sad to see that
Stephen. Just a quick comment on children with cancer. It is true that so far in the literature and also under our observation we have seen none. I can anticipate that I will be seeing one, which I think will be my first case of a child with cancer, and we will see whether GcMAF is feasible next week or in 2 weeks. But you're right, so far there are no cases described in the literature. The point is that until recently, most general practitioners or also most oncologists didn't know about immunotherapy with GcMAF. Please consider that many of the cancer patients that we are observing are self-administering the GcMAF, many times contradicting what their doctors tell them to do. So with children, of course, is much more difficult. We will be seeing maybe our first patient in 2 weeks. So maybe next year we will be able to provide us some more information.
Well, thank you very much, Professor Raed. And I'd just like to say a couple of things. If you decide to go for a Nagelase test, do not stop the treatment to wait for the results, because the results can take 6 weeks. And you need to get the blood centrifuge to take out the red blood cells before you post the blood sample to ELL. If you do not do that, the red blood cells rupture and the test is worthless. So you've got to find a laboratory to spin out the red blood cells before you start. So what you want to do is you want to take the blood sample, start the GcMAF, and then wait for the results. Okay, well, the next speaker is Dr. Rod Smith, PhD. He's our senior PhD who does the assays on the GcMAF. In all, there are 9 assays performed, and they have produced some very interesting scientific results on which papers have been written. Dr. Rod Smith.
Thank you, David. Thank you very much. I'm here from Macro Innovations, which we are based in Cambridge. We do the testing, as David says, for the GcMAF batches that we work with. My colleague Emma Ward is here with us as well. We're a 2-person company, fairly small, quite reactive in what we do. So if we have any questions, I don't think there'll be enough time after this, but please do stop Emma or myself and ask away. We'll do our very best to try and answer them. GcMAF, I think the professors Regaria and Raed before me will make this a little bit shorter, which is quite good. These are the names that we've heard this morning. It's quite confusing, as Professor Raed has said, that it comes under many guises, but the one that we're interested in, as you can see, is the actual math bit, which is made up of these letters. So it gives you an idea of what it is. Professor Raed has gone through this, and it shows you where the sugars are in terms of the actual enzyme activity to make MAF active. You've seen this just before, so I don't need to go into that again. And just to remind you, the action of Nagelase is just there, and it can't do this one over there. The testing is quite important, as we all are aware, mainly for the safety of the product. We want to make sure that it's safe for use. We want to know that it's active, and we also want to show that this week's batch is the same as last week's batch, which will be the same as next week's batch, and also for the people who are taking it. But that's very important because you don't want concentrations going up and down, which might cause aberrations in the actual treatment profile. So we do a range of tests. We do some biochemical tests. So they're the usual standard ones that you would expect. We do sterility. We commissioned external health to look at a sterility assay for ourselves, and that meets the European pharmacopoeial standards and the United States pharmacopoeial standards as well. One of the most important things is to make sure there's no endotoxin present. Endotoxin is a measure of bacterial contamination. We use an in-house FDA-approved system
which is calibrated. We have that calibrated regularly, and there are limits which are on that system to actually show how much endotoxin per dose you can have. And as far as I'm aware, we've not ever gone above that in any of the batches, so we're quite happy with the endotoxin testing that we do, and we're quite happy with the fact that it's safe in this respect. We do protein quantification, so we monitor the amount of protein in terms of the MAF that goes into that with a standard assay, and we also check the pH, and that's our biochemical tests. We do some identification because we want to make sure that what we've got and what comes to us for testing is what it should be. Is it what it says on the tin? We do the polyacrylamide gel electrophoresis. We do silver stain on that because the quantities, as you know, are very small. We also do an actual identity with a Western blot, so we do the gel, and then we also do the Western blot with the standard helix pomatia. And we also do a vitamin D antibody binding antibody test as well, so we can see that in the Western blot. This is an example of the blot that we do. You can see the samples, and you can see the identification, and there's a little bit up here which looks like it's dimerizing. It's not fully reduced, I don't think. So we're quite happy there's nothing else there. There's no cross-contamination. There's no other proteins that it binds to. So this is the sort of pattern that we're looking for in our tests. The major thing I think today we want to talk about is the activity assays. We do several activity assays to make sure that Gc-MAF batches are active. We do a proliferation assay, which is counting the number of cells, and we show that with an MTT assay, so it's a colorimetric assay, which is pretty standard and pretty robust. We use urine macrophages for that, the whole cells. We do a phagocytosis assay, so we actually monitor that under video, and I'll show you some of those in a minute. And we also look for morphological changes, so the phenotype actually changes from what we consider to be a normal standard cell in terms of the carcinoma way that it cultures to something which is more akin to the normal cell, which is not carcinoma. These are the results that we get from the proliferation assay. I think you can see here, this is the sort of standard that we get from the blank and the GC globulin, and then there's a second batch of internal standard GC globulin that we use. There's a sample that we test, and we also have an internal sample to make sure that we're positive in that. So the results that we get, we're fairly happy with our proliferation assay, that we definitely have activity. I've included this because this is with our collaborating group in the University of Florence. They use a different macrophage cell line. They use MM6, and it's interesting to know that their results in a slightly different manner are all very similar to what we would expect in the retardation of proliferation. And it's also quite good to include this one here, which is the Bravo probiotic, which hopefully somebody will get a sample of, because it shows that this is also active even in the yogurt. So on to the activation assays. The things that we're looking for, we're using these cell lines. So we use the normal standard MCF7, which is the human breast carcinoma. Almost everything that's reported on breast carcinoma starts with the sort of this cell line. It's kind of standard for the literature in terms of academic studies. So if you don't do it with MCF7s, people tend to don't believe you. We're also trying to culture other melanoma cell lines like human melanoma. So we have a cell line in MM127, which has a very different morphology and growth pattern to MCF7. Again, it's quite an important cancer line to look at. We've got some prostate cancer lines in, but we're culturing those up to look at them. We use two macrophage cell lines. One is the standard raw 264.7, which everyone knows about, and we've also obtained from the ATCC a human spleen macrophage, which is called 9853. The actual time-lapse photography, we did start trying to take a frame every 3 minutes, but the files were extremely large, so we now take every 3 minutes, and we culture up to 10 days. It's made on a standard microscope, and I'll show you a little picture of that. We get over the pH issue by using added EPs, which doesn't affect the growth or the interaction at all in some of the studies we've done, so we use 50 million on that. And we actually put the small